This week we were learning about the reproductive system, and so I chose to do my study aid on sex determination. On the front page of my slideshow, I show a very basic diagram of how sex is determined. So females have two X chromosomes, males have an X and a Y chromosome, and as we know, the sex cells are all haploid, meaning that they have half as many chromosomes as our other cells. So some of the male sex cells have the Y chromosome, and some of the male sex cells have the X chromosome, but all female chromosomes have the X chromosome. So as they combine, there's a 50% chance that two X chromosomes will combine to make a woman, and there's a 50% chance that X and Y chromosomes will combine to make male. So there are three sets of sex organs in both males and females. That's going to be the gonads, the internal genitalia, and the external genitalia. For males, the gonads are going to be the testes, and the testes produce sperm. For females, the gonads are the ovaries, and they produce either eggs or ova. They're the same thing. For the internal genitalia, this is just what connects the gonads to the external genitalia, and the external genitalia is going to be all the external sex organs that you can see with your naked eye. All cells but the sex cells have 46 chromosomes. These are considered diploid or 2N, and this just means that there's two copies of every gene, one from the mom and one from the dad. Sex cells have 23 chromosomes and are considered haploid or 1N. And this allows, once an egg and a sperm combine, they'll have 46. If they were both diploid and combine, every new generation would have double the amount of chromosomes that we need to be human. So sperm for males either have an X chromosome or a Y chromosome because the sperm is the sex cell. Eggs always have an X chromosome because that's all the female can pass down. Therefore, sex is determined by whether the sperm joining the egg contains an X chromosome or a Y chromosome. A lot of times in like monarchies, a long time ago before we knew that gender was determined by males, kings would execute their wives for not being able to bear sons. This is really awful, and now we realize that it was actually the king that was unable to bear the son. And this is just by chance, of course. Something that happens is abnormal distribution of sex chromosomes in the sex cell formation. Sometimes there will be an XX or a Y, and the presence of a Y always yields a male. And the presence of an X is essential to life. So an XXY would be a male. Just an X chromosome would be a female, but this female would have Turner syndrome and would not be able to reproduce. And the presence of only a Y would not be viable, so this would not form into a child. So as we know, anyone who we've ever known who is pregnant does not know the sex of the child immediately upon conception. You have to wait a long time, and this wait is often very exciting but very anticipated, and it's really exciting to learn what it is. So the reason that we have to wait to determine the sex of a child is because the sex organs do not begin to develop until the embryo reaches 7 weeks. And at this point, it only begins to develop. This doesn't mean that we can determine it exactly at 7 weeks. Prior to the determination of the embryo, it's considered bipotential. This bipotential gonad consists of an outer cortex with an inner medulla and two accessory ducts, the Wolffian ducts and the Mullerian ducts. The Mullerian ducts play a big part in female determination, while the Wolffian ducts play a big part in male determination. In the presence of the SRY gene, testes develop, and in the absence, ovaries develop. The presence or absence of this gene will cause the Wolffian ducts, if present, to develop, and the Mullerian ducts to not develop, or in the absence, the Mullerian ducts to develop and the Wolffian ducts to not develop. So the presence of SRY would cause male development. So the presence of this gene will cause the production of testes determining factor, which binds to DNA and activates the production of more proteins that lead to testes development. So once the testes are fully differentiated, there are 3 hormones that are secreted that play an essential role in the development of internal and external genitalia. The anti-Müllerian hormone is secreted from the Sertoli cells, and this causes the regression of the Müllerian duct. Testosterone and dihydrotestosterone are secreted from the interstitial cells. Testosterone causes the Wolferian duct to develop into the male accessory structures and controls the movement of the testes from the abdomen to the scrotum later on in the fetus development, while the dihydrotestosterone, otherwise known as DHT, causes the development of the external genitalia. In female development, the absence of the SRY gene causes the ovaries to begin to develop from the cortex of the bipotential gonads. So the Müllerian duct develops into the upper vagina, uterus, and fallopian tubes, while the Wolferian duct degenerates and the female external genitalia begins to develop because there is no presence of the dihydrotestosterone. Going back to the male development, we know that in the presence of DHT, the external genitalia is formed, and in the absence of DHT, there is no external genitalia, male external genitalia formed, and therefore female genitalia is formed. There's actually a syndrome that can affect fetuses in the womb, in which 5-alpha reductase deficiency is what it's called, and it's a condition that affects the male sexual development before birth. So what this is it doesn't allow enough production of DHT. Therefore, the male has a testosterone which develops the testes, but in the low amounts of DHT, they do not produce proper external sex organs. So something really interesting about the genetics of sex determination is in all of our cells except our sex cells, we have 46 chromosomes. That means 2 copies of each gene, one from our mom and one from our dad. Therefore, the patterns of recessive and dominant traits can take part, and we will most likely have the dominant trait of all the genes that we have, or most likely. So in females with X-linked genes, the dominant trait will always come about within us. But in males and with X-linked genes, they will always just take after the mother. There will not be a recessive dominant pattern going on. Therefore, certain diseases such as muscular dystrophy, hemophilia, and colorblindness are much more common in males because those are X-linked, and they will always follow the pattern of their mother. Their mother may not exhibit these traits because their mother has all their cells contain 2 copies, one with the color blindness gene and one without. Therefore, their mother will exhibit the non-color blindness gene, yet they can still pass on this gene to their children.